So InterMine's been around a long time. We started back in 2002 with the FlyMine project, which integrates Drosophila melanogaster and other Drosophila species, genomics and proteomics data. It's open source software, so we then found that other people started to use the software to integrate their own data. We had a big collaboration with the model organism database groups, and that has resulted in there being a yeast mine, a zebrafish mine, a rat mine, mouse mine, and a worm mine. And these intermines are still going. Over in the States now, you'll see there's a lot of plant-based intermines. Here in Cambridge, we also developed the human mine database. Over in Japan, there's a target mine which looks at drug target data, human drug target data. So there's lots and lots of intermines around now. If you want a fully comprehensive list of all the intermines that are out there with links and information about each one, then we have a registry that you can visit, registry.intermine.org. So, of course, each of these intermines have their own sort of data focus, but mostly intermines pull in data from other sources. So often the big database providers such as Uniprot and Intact. So we tend to load the genome annotation and sequence data, the gene ontology data, the large protein interaction data sets, and then each one also loads their own sort of types of data. So each intermine has its own data model, but we have the concept of a core data model which covers the main data types that are in all the intermines, so the genome annotation and sequences, the gene ontology, the pathways, etc. So you can go to different intermines and run the same query across different intermines quite easily. So why would you want to use InterMine when there's so many other databases already out there? So of course there's obvious advantages of having data integrated, different types of data integrated into one database. You can query across those different data types all at once without having to grab information from different databases and resolve the formatting and identifier issues that you would have. We can provide a nice identifier resolution system, being able to convert between different identifier systems and types and update old identifiers into the most recent identifier set. We can provide collated information about individual items or sets of items. And of course, it's a common platform, and you can see there's a lot of intermines, so it's a common platform for many organisms now and lots of different types of data. We have a very extensive API, as you'll see later, and you can also build your own intermine. Going back to this common platform, so having a common platform to so many different organisms means we've been able to look at interoperation between the different intermines. And so for the human mine and the model organism database intermines, you can seamlessly move between the different intermines. So for example, if you're in human mine, looking at your favourite gene, you can jump to your autologous gene in mouse or fly or fish very easily. And then of course, having the same interface means it's very easy for you to navigate the data in each of these intermines. A number of the plant intermines also are linked in this way. Okay, so you can build your own intermine, but we're not going to cover that at all on this workshop. At the moment, it takes some computer expertise to build your own intermine, but there's an awful lot of information available on the InterMine website, and a lot of help available, mailing lists and help pages, if that's something that you're interested in doing. But we also currently have a grant in progress, which is making InterMine available to the lab researcher. So this is looking at making it very easy for someone who's generated their own data in the lab, who isn't a computational or bioinformatics expert, to add their data to an InterMine on the cloud, with the advantage, of course, that they can integrate their data with additional data and take advantage of all the analysis and visualization tools that are available in InterMine. This is important because there's such a big move towards making data fair nowadays. So this is once you are ready to publish your data, you need to make sure that that data is findable, accessible, interoperable and reusable. And so this is one way of researchers being able to facilitate that. Okay, so before we move on, I just want to do a quick poll. So I'm going to, if you go to the, hang on, on the forum, I just want to get an idea of how many of the participants on this workshop have used or either never heard of InterMine, heard of it but not used it, or have used it? Okay, brilliant. I think we are pretty much got everyone there, so I can share the results. We have 75% never heard of, 19% heard of but used, and 6% have used a little. So that's perfect, because this is very much going right in at the beginning, completely new to InterMine. So hopefully you'll all learn something useful today. Okay, so I'm going to go back to my slides. Daniela, you can see the slides?
Yes.
Yeah, it's all good. Okay, so before...
Sorry for the delay. I was unmuted myself, sorry.
Okay, thank you, Daniela. Okay, so just before we move on to actually looking at the InterMine user interface, I'll just talk just a little bit about InterMine accounts. So you can go to any InterMine and use it without creating an account. It's all free, it's all open source. But if you do create an account, it's very simple, you're just giving a username and a password. That allows you to save any searches and lists that you create permanently and do nice things like share lists of genes and proteins, etc., with your colleagues. At the moment, you do have to make a separate account for each InterMine database, but we are working to make that easier. So if you are following along actually on an InterMine today, you might want to just quickly create yourself an account so that anything that you do is saved. Okay, so we're going to move on and start looking at the user interface. So this slide shows the sort of main features of InterMine. So you tend to come to InterMine either with looking up something about a single gene or a particular protein, or more commonly nowadays, you may come in with a list of genes or a list of transcripts that you want to analyze. In the center panel, we have the main search interfaces that are available. So these range from the simplest level, the keyword search, through to some predefined template searches, a more advanced query builder, and something called the region search. And then the data from these searches is presented to users either as report pages, list analysis pages, or results tables. And we're going to take a look at each of these in turn. We'll start off at the simplest level, looking at how you can come in, looking at a single gene through the keyword search, and take a look at the report pages. So the keyword search, it's a faster keyword search. You can make a very specific search, for example, search for a specific gene, for example, Paxt6 or PPARG, from where you can access the report pages. Or it could be a more exploratory kind of search. You could search with keyword, for example, insulin or diabetes. And from there you can access the report pages or perhaps make a list of related items. And you can filter the results by data type. For the report pages, so everything within an InterMine has a report page, whether it's a gene, a protein domain. And the report pages collate all the data that we have available for that object. I'm sure you're probably all familiar with report pages from other databases. The data is presented in a mixture of interactive tables, research results and graphical displays, and there's links through to all the related data. So it's a nice way to start at one point data and explore through to other related types of data. So throughout the workshop, we're going to focus on the Paxt6 gene for our examples. So Paxt6 is a very highly conserved transcription factor. It's important in eye and brain development, and it's very highly conserved right through from human, through mouse, the fish and the flies, you can see from this slide. Okay, and so you'll see from my slides that I've left the exercises up there. I'm not going to follow them quite exactly, but if you want to go back and do some of the things we've done, then you can go back to these exercises. There's also in the link, there's a link to something called exercises with answers, which has screenshots showing you the steps for every single exercise, so you can follow that through afterwards if you wish to. Okay, so I'm going to go now to hopefully, to the HumanMine database. If I can find it on my share screen. No, not there. Where's it gone? Hang on. I'm getting
I see you, my home there. Okay.
I didn't see it. Where is it? Hold on one sec. Let me do the share again. New share. HumanMine. Found it. There we go. Okay, so hopefully now you can all see the HumanMine homepage. Okay, so before I do the keyword search, just take a look at this homepage, and you can see along the top the set of tabs. And these, of course, relate to some of the features that I just told you about. So we have template searches, using lists, the query builder, the region search. Every InterMine should have a data sources page, and this lists all the data that is integrated into that particular InterMine, including the versions, so you can see how up to date the data you are querying is. A link to the API, and then there's something called MyMine, which is where you manage your own account if you are logged in. So this will show you any of your lists and searches, and where you can manage your password and other account details. Okay, so I'm going to go to the quick search, which is in the top box here, and I'm going to search for Pact 6. Okay, so you can see the search results, and you can see that this search has taken Pact 6. It hasn't just searched for the Pact 6 gene, it's searched everything in the database, and it has found a match to 869 publications that mention Pact 6, 16 genes, and some other things. There's a protein domain which mentions Pact 6. You can see also that there's hits for 3 different organisms. This is because in HumanMine we not only load human data, but we also have some genes there for mouse and rat for the ortholog data, and some mouse allele data, for instance. So you can use this to, excuse me, to filter the data further. So for example, I could be interested in the Pact 6 publications. So if I filter to just one type of data, such as publication, I could then create a list of all the Pact 6 publications, for instance. So we're going to look at lists later. For now, we are interested in looking at the Pact 6 gene, the human Pact 6 gene, and that's the first link here. So I'm going to click on the link there. And that takes us to the Pact 6 report page. So all the report pages have the same basic structure, same type of structure. They start with a summary about the gene. So there's a description for the function of the gene, and there are all the different identifiers by which this gene is known at. Of course, for human, that's quite complicated. We have the gene symbol, the NTB ID, the ensemble ID, and the HGMC identifier, and all the synonyms that this gene has been known by. You can access the chromosomal location, and the FASTA link here will allow you to view and download the sequence for the gene. The next bit is a sort of a summary of the data that's available. This is actually specific to HumanMine. You won't see that on the other InterMines, but it just summarizes the pathways and diseases, etc., that are found on the report page. Looking further down, there's a set of quick links where you can quickly navigate to the different types of data on the report page. The first set of data you can see, for example, is some curated comments that we brought in from the UniProt protein database. So this is high quality curated data giving information about the diseases this gene is involved in, its function, its tissue specificity. I'll scroll down a little further, and we have some expression data. Excuse me. So this is data that we've pulled in from the Protein Atlas project, and it is protein localization data based on antibody staining. And as you'd expect, we can see that Pact 6 is highly expressed in the brain. And also here we can see that it's also, you see this coloured red for high expression, highly expressed in the pancreas. And some other types of data, so if we click on the disease quick link, we come to a table summarising the diseases that this gene is involved in. This is data that has all been pulled in from the OMIM database. And as you'd expect, we can see here a lot of the diseases that Paxt6 is involved in are eye diseases. We have an interaction viewer. So Paxt6 is a very highly connected gene. It interacts with an awful lot of other genes. Here you can scroll between physical protein-protein interactions and genetic interactions, for which there's only actually one for Paxt6. And you can select nodes to bring up further information about that particular interaction with Paxt6. You can also toggle on a table, show the interaction data in table form. So these tables are highly interactive, and we're going to go over those later. So you can be able to explore the data even further through one of these tables. One more thing, I'm not going to show you everything on the report page because you can explore these yourselves. We could look at the protein data, for example, and you could link through to the report page for the Paxt6 protein. And again, you can see similar structure, the main identifiers, again, the curated comments for UniProt. And if you scroll down, there'll be some protein specific information. So for example, here we have information about the protein domains that the Paxt6 protein has. This has been brought in from Interpro. Okay, so going back to so you can see the report pages contain an awful lot of information. It's presented to you in various different ways, depending on the type of data. Excuse me. Just going back to the Paxt6. Okay, and so the other part of the report pages I want to show you is in the top right-hand corner. So the first thing you can see here is something called lists. So we're going to look at lists in more detail later, but you can create your own list in an InterMine, but also a lot of the InterMines will provide something called public lists. So these are interesting sets of genes from publications or other sites that we provide for you to use. And in HumanMine, you have the Genomics England gene panel lists. So these are highly curated gene sets. So they're sets of genes where there's an awful lot of evidence that that gene is involved in the disease that that list is about. So we can see here that Paxt6 is actually found in a lot of these Genomics England gene panel lists, and obviously a lot of them are eye development abnormalities. And then further down, there are the links to other mines. So you'll remember in one of my slides earlier that I said you can seamlessly move from one InterMine to another. So for example, this is how you can do that from a report page. So for example, from here, we could click on Paxt6 underneath the MouseMine link there, and that will open up the MouseMine InterMine database and show us the report page for the orthologous mouse gene. And you can see this report page, same structure as in HumanMine, so easy for you to use and navigate. Some of the data may be presented slightly differently. There may be different data, but it's the same format, so it's easy to navigate. Okay, so I'm going to go back to my slides now. I hope. Keep losing them. Give me one sec. Okay, so going back to these slides, so we have looked at how you can come in with a single item, use the keyword search, and explore all the information within an InterMine for that thing, item, that object, that protein or gene or whatever it is you've looked up, and also navigate to related data and also to other InterMine databases. So what happens if you have a list of genes? You don't want to go to each, you don't want to put them in individually in a keyword search and look up report pages for each gene. You want to do that in bulk. So the first thing we need to look at is how can you get a list, your own list of data into an InterMine. So we'll look at the list upload, and then we'll look at these list analysis pages that are similar to report pages. So I'm now going to go to for the list upload, I'm going to use the Lyne database, just because we have a nice list upload example already set up here. So as you can see, this is the Lyne InterMine that you can see now. Notice the same tabs along the top. This time I'm navigating to the Lists tab. And under the List tabs, you'll see there are two subtabs, Upload and View. So first off, we want the Upload tab. So when you create a list, you can only create a list of one type of data. So you can't mix up genes and proteins or genes and transcripts. It has to be a list of all genes or all proteins, for example. So you first select your data type, although if you don't, InterMine's quite clever at converting things that you put in that aren't of the right type, as you'll see in a minute. It's a very good idea to select the organism, particularly if you're uploading gene symbols, because gene symbols are not unique between different organisms. And then you can either type or paste in your identifiers, which can be one per line or comma separated or space or tab separated, or you can upload from a text file. I've got an example already available to use here, so I'm going to click that, click to see an example here, and that's going to load up a set of example identifiers. And then when I create my list, it's going to compare all the identifiers that I've given it against the Lyne database and tell me if it's found a match for each identifier. So what we have here is it says you entered 27 identifiers, we found 25 genes. So there's been some discrepancies. And this page is going to tell us what those discrepancies are, so we know what we have in our list and what we need to do. So the first discrepancy it's found is some duplicates. So this often happens between genome annotation, gene model releases for organisms. Sometimes a gene will become split, so it is now two genes, for example. So here you can see the identifier we had in our list is E2F, but actually in the database it is found E2F1 and E2F2. So here we have to decide which of those two genes it is that we actually wanted, or both. And here we have to add the one we want to our list. Scrolling down a bit further, okay, so we still have some discrepancy because it said 27 identifiers, but it's only found 25. So here's a summary of the other things it found in its search against the database. So first of all, our direct hits. So most of the identifiers we gave, it found a direct hit in the database, so there's no problem. We can see one flagged up here. So this is where it's found two different identifiers that match the same gene. So we have a gene symbol and a FBGN identifier, which is the main fly base identifier type for fly genes. So that's fine. So we don't need to do anything. It's just that two of our identifiers match the same gene. We have another tab here called non-gene identifiers. So here we can see that one of the identifiers we gave was twist_drome. So this isn't a gene identifier, this is actually a uniprot protein identifier. But it's okay because InterMine has found the gene for that protein and it's automatically added that to our list. So we don't need to do anything again, if that's the gene that we want in our list. And then there's a tab called synonyms. So this is synonym matches. This is the most common type of discrepancy you'll find between genes that you upload and genes in the database, because gene identifiers do change quite a bit between genome annotation releases. So here, again, we don't need to do anything because if it finds a direct match between the identifier you've provided and a synonym in the database, it just updates the identifier to the correct one, so the up-to-date identifier, and adds that to your list. So the only time you need to do something is when there are duplicates found, unless of course you're unhappy with the automatic changes that InterMine makes. So once we're happy with our list, we simply give it a name. So there's a default there, but we can change that, call it whatever we like, and then we save our list. And once we've saved our list, you can see it's automatically brought us to a page. Now this is what we call a list analysis page. I'm going to flip back to HumanMine in a minute to show a list analysis page in a bit more detail, because this list of 25 genes actually isn't all that interesting to show in a list analysis page, but any list you upload, you will automatically get one of these list analysis pages, and they're automatically generated. If we go to the View subtab here, you'll see my list there at the top, so you can easily navigate and find your list again, and that link in my list will then take me back again to that list analysis page that you just saw created. So I'm going to go now back to HumanMine, and I'm going to navigate to the List tab of HumanMine. And at the top here you can see I have a public list called PL-Pax6 target. So this is, you remember I said Pax6 is a transcription factor, so this is a set of target genes, so the transcription factor. Does that load up? Okay, so list analysis pages are kind of analogous to the report pages, but rather than collating all the information available for one type of object, you now have the information for all the objects in your list. Any list as I've just said, I think any list that you create, whether that you upload from an external source or that you create from a query, as you'll see later, you'll automatically get one of these list analysis pages generated for that list. They all have like the report pages, they all have the same type of structure. So we start off with a table of all the genes or proteins or transcripts, or whatever type of object it is that you're looking at. So a table summarizing what is in your list. This is an InterMine, what we call InterMine results table or IM table. And we're going to look at these in more detail a bit later, because there's a lot of features for these tables. To the right of the list analysis page, you can see first off there's something called convert to a different type. So here I can convert my list of genes straight into another table for the equivalent proteins. This feature is particularly useful if you've done a transcriptomics study and you've actually uploaded a set of transcripts, because you can convert that set of transcripts straight into the equivalent genes, which tend to be most useful because most data hangs off genes rather than transcripts. So if you've done a transcriptomics study, you tend to want to convert those transcripts into a set of genes. Below we can do we have sets of orthologs. So the first section here called orthologs will provide you with a table of the orthologs within the HumanMine database. So you're not jumping out to another database here. This is just providing a table of the orthologs for a number of different organisms within the HumanMine database. As for the report pages, you can also link out to the other related model organism databases. So I could click on the MouseMine link here, and that will open up MouseMine with a list analysis page for all the orthologous genes for the genes in my list. Going back to the HumanMine list. Okay, scrolling down the list analysis page, we come to one of the, what we'd say, probably one of the most used features of InterMine. These are the widgets. Not quite sure what's happened to that one, but so here we have lots of enrichment statistics provided. So I expect everybody's heard of gene ontology enrichment generally, but we also provide enrichment tables for other annotation types. So in HumanMine we have gene ontology enrichment, protein domain enrichment. We do have publication enrichment, but something's gone wrong there, I'm not quite sure what, and pathway enrichment. If you go to MouseMine, for example, you'll find other enrichments provided. So there you'll get gene ontology enrichment, mammalian phenotype ontology enrichment, anatomy enrichment, disease ontology enrichment. So depending on the InterMine, you get different enrichment sets shown. For the enrichment, we use the hypergeometric distribution, and we automatically apply a test correction. And you can see here you can toggle different types of test corrections depending on how stringent you want it to be, or you can take off a test correction, which isn't necessarily recommended. The p-value applied by default is 0.05, but again you can change that. This is gene ontology enrichment, so you can toggle between the different gene ontology ontologies, so for biological process, cellular component, and molecular function. And quite importantly here, you can also change the background population. So by default, the enrichment statistics will be calculated against all the genes in the genome that you're looking at. So in this case, it's the human genome that have that particular annotation. So in this case, all the genes in the human genome that have a gene ontology annotation. That isn't always the background that you want. For example, if you've done a particular screen or used a particular microarray, for example, you want your background to be just the genes in that particular experiment. So if you upload that set of genes as a list, you can then change the background population here by simply selecting that list, and it will recalculate the p-values based on that set of genes rather than all the genes in the genome. Okay, the actual table of enriched terms is probably fairly self-explanatory. So here our most enriched term for this gene list is regulation of transcription by RNA polymerase II. The p-value is shown here, and then we have a matches column. So here we can see 150 of the genes from our list are annotated with this Go term. We can click on that, and from there we can view a table of just that set of 150 genes. So this is a nice way that you can view subsets of your data and also create sublists of your data. So you could create a list, for example, of just those 150 genes. So this is a particularly used feature of InterMine. A lot of people come in to use the enrichments that we provide. All the tables have the same structure, so I won't go through another one. And then down at the bottom of the list analysis page, we have something called, these are pre-run template searches. So we'll look at template searches in more detail in a little bit, but basically they are predefined queries that are automatically run on the genes in your list. So for example, here we can quickly get a table of all the human phenotype ontology annotations for the genes in our list. Okay, so that is a lot of information about lists and list analysis pages. So going back to our slides. So we've done uploading a list analysis. Okay, so we've pretty much covered how you can explore, using InterMine to explore data for a single item or a list of items, either by exploring through the report pages or by creating a list and looking at the list analysis pages. So now we're going to look at more sort of detailed analysis and how you can ask more specific questions about the integrated data. So first off, we'll take a look at template searches, and through this we'll also take a deeper look at the results tables. So the template searches, these allow a more refined search from the keyword search and the report pages, but they've been designed so that they're quick and easy to access. So you can come into an InterMine and not have to learn how to create a query as you do with our query builder. You should be able to just come in and run the search that you want. They're basically predefined, a simple set of filters with default values. They range from simple searches, which may cover just one data type, and to more complex searches that can span several data types. And pretty much every template you can run with either a single item, for example, a single gene, or with a list. The results are returned in the results tables that we'll have a look at. And the nice thing about template searches is that they're very easy to add to an InterMine. So if you're looking at an InterMine and you don't see a template that you want, but you feel like the data is there, that you should be able to, run the search that you're wanting, then you should ask the InterMine provider if they can provide that template for you, because they can be added within minutes. you don't have to wait for a new database build or anything like that. That's a way we often provide help to our users. If someone comes to us with a question, I can quite often return by saying, oh, try running this template, and provide them with a template search. Okay, so most InterMines have a very big library of template searches that cover all the different data types within the InterMine. So we'll go now just to have a little look at what these templates look like. Okay, so I'm now going back to the HumanMine database. Scroll to the top and I'll go to the templates tab. Okay, now you can see in HumanMine, there's a pretty big long list of template searches there. So how do you find the template that you want? Well, you can filter. Say, for example, if I'm particularly interested in genome-wide association studies, I could put GWAS there, and that will filter that list to a reasonable number of templates where hopefully I'll find the type of search that I am looking for. There's also a category filter. So you can filter, for example, to all the expression templates. If you're logged in, there's also the option to mark templates as favorites. So if there's particular templates that you keep coming back to, you can star them and then you'll be able to more easily find them when you come back to the InterMine. So all templates, the same sort of structure, have the same basic structure. They have, so let's look at this top one as an example. It's called gene to pathway. So it starts with an input class. So we're starting with a gene, and for that gene, we want to return the pathways for that gene. So they always have a short title and a longer description, which quite often also lists the data sources that will be returned. And you can see this example, it's a very simple form. We always provide a default because that makes it easier for you to see what sort of values are expected. So here you can add a single gene identifier, or you could select a list of genes to run this template on. Looking at maybe giving an example of a slightly more complex template. There's one here called gene plus tissue expression to interactors that are expressed in this tissue. So this is a nice example of how by integrating the data, you're able to ask more sophisticated questions across the data, because here you're taking the gene, tissue expression data, and interaction data, and in a very simple form, you're asking a question across those data types. So in this case, you enter a gene and a tissue, and it will return any genes that interact with the gene that you've given that are also expressed in the tissue that you specify. So a very simple form, but actually quite a complex query that would be very difficult to ask if you didn't have this data integrated in the way that we do in InterMine. Okay, so I'm now just going to go back to my slides. Okay, so the next part of the workshop, we're going to be sort of working up through, what I'm going to show you is sort of working through a sort of a use case. So going back to our very first look at Paxt 6, when we did the keyword search. So we know that Paxt 6 is involved now in brain development, but we also saw from that protein atlas data that Paxt 6 was highly expressed in the pancreas. Now this may have come as a surprise to us. We may not be experts on the Paxt 6 gene. So we may want to use the template searches to find out more information about this role of Paxt 6 in the pancreas. So we can use the template searches to ask questions like that, like what other genes are involved in pancreatic function, or are there potential targets of Paxt 6 in the pancreatic tissue?
Rachel, sorry to interrupt you.
Yeah.
Before starting the second part, there are 3 questions on the OpenAI platform.
Okay.
One involve, starting from the bottom, one involve, I think, the enrichment. If you can go through the OpenAI platform and see.
Yes, sure.
So yeah, it says, is it possible with InterMine to connect to expression data to perform, I think it is a gene set enrichment analysis with the expression data set. So this is more related to enrichment, I think.
Maybe I better just read the. Is it possible with InterMine to connect to expression data? Not at the moment, if I understand the question correctly. I mean, generally, if you have expression data, you could take your differentially expressed genes and do enrichment analysis with those. Maybe I don't quite understand the question exactly. So you could take a set of differentially expressed genes, upload them, and do enrichment analysis on those, or not even differentially expressed genes, any expression set. But maybe if you want to elaborate on the question a little, I can answer it a bit better. But at the moment, there's nothing specifically connecting expression data and the enrichment statistics. Okay.
And there is another couple of questions, Rachel. Have you seen?
Yeah. Okay.
Or do you want to keep?
I might answer the others at the end as they're more general. If that's okay. I'll keep going. Okay. Okay, so we're looking at the template searches, and we're going to do a search to try and find out a bit more about the role of Paxt in pancreatic tissue. So our next sort of exercise or demonstration, I'm going to go back to InterMine. Use InterMine. Okay, and I want to find a template that will allow me to query the protein atlas data specifically. So I'm going to type protein atlas here, and the first one I can see is gene to protein tissue localization, and we're going to use the set of Paxt target genes that we looked at earlier. So we're going to constrain our template here, and we're going to scroll down until we find, there's quite a lot of lists here, the Paxt target set. And then to run the template, we simply show the results. Okay, so what I'm going to do now is use this template results just to explain a little bit more about the results tables. So let's try and understand what this template has given us. So we've put in a list of genes and asked it to return expression data from the protein atlas data source. So our first column is the gene primary identifier. So this is an NCBI gene identifier, and this is the corresponding gene symbol. We then have the cell type for the protein atlas expression. We then have a column showing the level of expression, reliability score that is provided by protein atlas, and then a set of tissues. So the protein atlas data gives tissue data, but you can also do much finer granularity by going actually into individual cells within that tissue. Okay, so, and you can see along the top of each column there are some icons. So most of these are probably fairly self-explanatory. Just to whiz through these, you can sort on a particular column, you can remove or toggle columns. And then the last two are the ones which are more useful. You can add a filter to a particular column, or there's something called the column summary, which is the feature that I use and find the most useful. So if I go to the last column, the tissue name, and I click on the little graph that shows me the tissues, I can use this to filter. So we're actually only interested in the pancreas. We're not interested in the all these other tissues. I can use this column summary to type pancreas in there to actually filter the table to just the results for the pancreas. I could have also done that by specifically adding a filter, but generally I find it easier to use the column summaries for this. Okay, so now also if I go to the expression level column, now we can see we've got 431 rows of data and a number of different expression levels. We want to know how many different expression levels there are. We don't want to have to scroll through 431 columns to find that out. So if we use the column summary again, we can immediately see here there's 4 different levels available throughout these 431 rows. So there's not detected, medium, low and high. So for this we are only interested in genes that are actually expressed in the pancreas. So we're going to select medium and high, and again filter our table again to just genes from that list that are expressed at either a medium or high level in the pancreas. We're not going to worry about the different cell types for now. So let me now just show you a little bit more about the functions along the top here. So we have something called manage columns. So if you click on that here, you can use this just to move around the columns or delete them if you want. But quite usefully here, you can add a column. So just so this shows the gene class, the attributes of the gene class, and you can scroll down and access all the other classes related to genes. So it's sort of like building a query. You can add a column from any other class. It may not make sense if you had something silly, but just an example, I will select the gene length. Add one new column, apply the changes, and you can see, and you can see at the end of my table, no, hang on, I just did something really silly then and clicked on something. I'll just do that again, add a column, gene length, add one new column, apply changes. Okay, it's added the gene length as another column. Okay, gene length maybe isn't the most exciting feature to add, but I can just show you now that if you have numerical values, the column summary gives you the distribution of those values rather than the other type of column summary. So it'll give you the minimum, maximum, standard deviation and average for the values in that column. You can also add columns and change things by navigating to the query builder, but we'll look at that a bit later. Then there's a manage filters box. So this allows you to manage the filters that are applied on any of the columns. Hang on, I've lost my filter. I think when I just let me add those back on because I just somehow navigated away and lost them. Okay, so when I click on manage filters, you can see there are all the filters that I have applied. So we have a column we have a filter in place because we have only run this query on our Paxt target gene list. We have filtered this, the tissue name column to pancreas, and we also put a filter on the expression level column of just medium and high expression. So we can edit all these filters or remove them from here. As we can, if we're just interested in one column, then we can access the filters on individual columns as well. So the manage relationships, so this is a slightly more complicated feature. So you probably noticed that as I've added these features to the table, the number of rows, the table's got smaller. So we've been reducing our data set, filtering it down according to the filters that we've been adding. You can also change that so that the data is not reduced. So for example, I've got my set of genes. I want to know if it's expressed in the pancreas. There's 2 ways I could return that table. I could return all the genes and show if it is expressed in the pancreas, or I could do as we have done here and only return those genes that are expressed in the pancreas. So it's a way of managing how you want that data to look. the default way is to reduce the data, so that when you add a filter, it reduces the table down. But you can change that and show the table in a different way. I hope I've explained that okay. Okay, over on the right here, a couple more features of the result table. I'm going to come save as list in a minute. Generate Python code. Daniela will talk about this to do with the API, so Daniela will talk about this later. You can export any data as you would expect in a number of different formats. So you can export your data tables as tab or comma separated values in XML or JSON format. And you can also export the sequence data. So for example, you could export the FASTA sequence for the set of genes in your table, or also as GFF3 or BED format. You can play around with columns and rows and compression and however you want your table to look when it's downloaded. You can also send data straight to Galaxy. So if you're familiar with Galaxy, it's a bioinformatics platform with a lot of analysis tools. So you can send data straight from InterMine to the Galaxy platform. And finally, save as list. So this is a really nice feature. So here we started off with our gene list. We have returned for those genes, those that are expressed in the pancreas. And now if we want to carry on analysing that set of genes within InterMine, we want to save that set as a list. So we can save out our 129 genes, and we can call that Paxt targets expressed in pancreas. We can save that set as a list. And we're going to be looking at how we can further analyse that list as we go through. So it's created a link here. If I click on that link, it's going to take me to the list analysis page for the list I've just created. And if I want to find that list, I can see it's created there. Okay, so just going back to my slides. Okay, so we've looked at results tables. I've hopefully shown you how useful those column summaries are. And so the next thing I want to look at is the query builder. Okay, so this is our more advanced part of the InterMine interface. It's our custom query builder, but it has the power that it does allow you to create and save your own searches. So unlike the templates where you're sort of limited to what we've created, you can combine any classes of data that you want and apply your own filters. You can also use the query builder to modify template searches. So it takes a little bit more learning than just coming in and running a template search, but it's easy if you remember there's 3 steps to construct a query. First of all, you navigate the data model to find the class or attribute that you need, and then you apply the appropriate constraints to those classes or attributes, and then you just decide what columns that you want to view in your results. So we're going to go to HumanMine again. And I will go to the Query Builder tab. Okay, so the first thing you need to do when building a query is decide on your starting class. So you remember when we looked at the templates, there was always a starting class and then the output. So it's similar to that idea. Where do we want to, what's our sort of input to our query? We'll start from gene. Okay, so what we're going to do is build a query to return genes from humans. We look at the disease data and return any genes that are involved in the disease diabetes. Obviously this is related to pancreatic function. So this is the query builder. So there are basically 3 parts. This is the model browser in the left pane. There's a query overview in the right, which you'll see in a moment. In the bottom, we build up the columns that we want to see in our results. So you'll see in the model browser, we've started from the gene class. So all classes have attributes. So genes, of course, have a primary identifier, a symbol, a length, etc. And the gene class, of course, is related to other classes of data. So genes are related to the allele class, the disease class, for instance. So you can navigate from the gene class to all other related classes. We can expand the allele class, for instance, and you'll see attributes for the allele class. And further down, you can see alleles, of course, have genotypes, so you can navigate to the genotype class. So you can navigate right through the data and select the data classes that you are interested in. You'll see after each class and attribute, you can see two, hopefully you can see this clearly enough, there are two little boxes. The blue one says show and the red one says constraint. So this is where we define our filters and our columns that we want in our results output. So to start off with, we're in the gene class. We want to see the primary identifier and the symbol. So I've just clicked on show next to primary identifier. I'm going to click on show next to the symbol. Now you can see here there's building up on the right hand side, a summary of what I've done. I've started from gene, I've clicked on primary identifier and symbol. They have appeared as boxes down here. So basically these boxes will represent columns in the results table. So yeah, so we have a primary identifier and symbol so far in our results. Okay, as this is HumanMine, as I said at the beginning, even though it's based on human data, we do have genes for mouse and rat in the database as well. So we do need to put a constraint on the organism as well, or we may get genes for mouse and rat returned as well. So I'm going to navigate to the organism class. I'm going to put a constraint, click on constraint on organism name. And here I get a drop down list. I'm going to select human and add that to my query. And you can see that constraint has appeared on the right here. Okay, so if I run the query now, I would return all genes from humans. But actually what I want to do is navigate to the diseases class. So the diseases class are data from the OMIM database. I want to, in my results, see the disease name. And again now, I've added the disease class, but I haven't added constraint. So if I run this query now, show results, I will return all genes from humans and all the diseases associated with all genes. That's a little bit more information that I actually want, so I can navigate from my results table, from the trail at the top here, back to my query, and I can add a constraint to the disease class. So I'll click on constraint next to disease name. And here I'm interested in diabetes, but there's lots of different types of diabetes. So I don't want to put a constraint equals diabetes because I probably won't get anything back. I want to make it a less strict constraint. So I'm going to select contains and put the word diabetes in. I could, from the drop down, select a more specialized disease name if I wanted to, but I'm going to leave that as contains diabetes and add to my query. Okay, and so I'm going to run that query again. And now I have all genes from humans that are involved in some form of diabetes. For this example, I'm just going to leave it like that. But of course, I could use my column summary to see how many genes there are involved in each different type of diabetes. So I can immediately see I've got 28 genes here involved in diabetes mellitus, non-insulin dependent. So again, the column summary, a nice way to look at the distribution of your data. I'm going to save a list of my genes involved in diabetes. I'll call it human diabetes genes and create that list. Okay, so that's a very sort of quick view of the query builder. Hopefully that wasn't too quick. Go back to my slides. Okay, so we've covered nearly all aspects, admittedly very fast, of the web interface. We've covered the keyword search, the template searches, the query builder. We've looked at report pages, list analysis pages, the results tables. One thing I haven't shown you yet is the list set analysis, and I find this feature, this is very useful if you want to build up a more complicated analysis workflow using the interface. So I'm going to go back for our last exercise. I'm going to go back to HumanMine to show you just one last little bit. So we created two lists. So first off, we started off using the template searches for our Paxt target list. We found out which of those genes are expressed in the pancreas. Then using the query builder, we created another list which gave us human genes that are involved in diabetes. So now I can see the two lists I've created here on the lists page. I'm going to select those, and you can see the set operations that are available to me are at the top here. I'm going to select intersect. Why isn't it working? Oh, there we go. That's working. Intersect. I need to give it a new name. So this will be Paxt genes expressed in the pancreas that are also involved in diabetes. I can save that set, and it gives me 3 genes. So we've got 3 genes that are Paxt targets expressed in the pancreas that are also involved in diabetes. And of course I can explore these 3 genes further. There's only 3 genes, so I can look at their report pages, I could look at the list analysis page, or I could feed that set of 3 genes back into, for example, another template. I may be, for example, interested in genome-wide association studies. So I could find gene to GWAS hit is a template. I could select that list that I've just made. What did I call it? That is down at the bottom. Paxt genes expressed in the pancreas involved in diabetes. So all these. Okay, so this is a large number of genome-wide association studies. So we can see that we don't know how many of the genes in our list have been returned, so we can use the column summary, and we can see just 2 of the 3 have genome-wide association data associated with them. We can take a look at the results phenotype in the column here, and that shows us, okay, there's some studies which looked at type 2 diabetes. We could select that. Oops. There we go. So we can see, for example, those 2 genes, the same 2 genes, have also been shown to be involved in, have some involvement in type 2 diabetes, or have the SNP associated with them through genome-wide association studies. Okay, so what I was trying to get across from that last section was that although I've shown each feature of the interface individually by looking at templates and queries and uploading lists and creating lists, everything is interconnected. So you can do iterative workflow analysis. You can run a query, you can refine it, save a list, combine it with another list, plug that into another query, for example, create sublists through the enrichment statistics. So everything's interconnected, and you can build up some quite nice analysis workflows. Okay, let me just check what time it is. 5 past. That's great. 10 past. Okay, so one very last bit of the interface that I'm going to show you very quickly before we stop for questions is the region search. So the region search allows you to search for features that overlap a list of genome coordinates. So you basically add your genome coordinates and it will show you any sequence features, for example, genes or regulatory features that overlap those sets of genome coordinates. And the interface allows you to extend those regions on both sides. So you can, if you have, for example, a set of SNPs, you could extend the area around those SNPs. Or if you had a set of transcription factor binding sites, you could either put them in as they are, or you could extend them on either side for your search. So Sorry, I'm going back and forth here between the slides. This is the last time, I think. So back to HumanMine again. So of course we go to the regions tab. Actually, I'm going to use Lyne. Just so that, where did I go? Hang on. No, I'll do new share. There. Where's that one? No. Excuse me a moment. Doesn't seem to want to share the right thing. Oh, here we go. Okay. Give me 1 second. I'm not used to doing these things virtually. I can't find my tab at the top. Hang on a moment.
I see the MouseMine tab next. Yeah.
Yeah, I was going to use FlyMine, but I can't get to it. Hang on a minute. We can move. Is it that one? Oh, it doesn't matter. I'll use HumanMine. It's fine. Okay. So the first thing is select your organism. So we only have human available in HumanMine in this case. And then you want to select the features that you're interested in looking at that may overlap your genomic regions that you're going to supply. So there's an awful lot of different types of sequence features, but we will just select genes perhaps, and what else shall we select? Maybe some gene flanking regions, exons. This is why I was going to use FlyMine. Okay, and then you put in your genome coordinates. So I've got an example here. So quite commonly genome coordinates are written like this: chromosome, colon, start, end. But you can also put these in just as tab separated, chromosome, start and end. If you're putting in SNPs, even though the start and end is the same, you still need to put in the start and the end coordinate. You can also upload your set of genome coordinates from a file, of course. And this is where you can extend your regions. So you can either write something in the box or extend using the slider here. It will only extend both ends. You can't do it one end or the other. And you can also select a checkbox to do a strand-specific search, if that's what you want. And then we select search. Okay, and so the table that's returned, it provides the information for each region that you've put in. So you can see here, this is the first region that I put in. This is the feature that it's found, the feature type, and the location of that feature. As we scroll down, you'll see the information for the other regions that I had in my example regions. What can we do with this information? We can work with this information either for each individual region or for all regions as a whole. So we could export the data in various different formats for all of our regions, or create a list, so we could create a list of all the exons, for example, that overlap our regions. Or we could do the same individually for each of our individual regions. And we can explore each feature. These will link through to the report pages for each of those features. So there's a gene, for example, overlapping one of our regions, and there's the report page for that gene. So fairly simple. That's the last feature I wanted to show you of the InterMine interface. And now we will go to questions. Okay, so I need to go to the do we have many questions, Daniela?
A few of them. Maybe we'll start from the most popular, which appear on the top.
Okay, So that says, hi Rachel, great tool so far. I was checking around in the HumanMine regions sheet, selecting human as organism. It doesn't show the genome build. For the ones of us that deal with annotations, transcriptomics is an important detail to have to know the versioning. Okay, yes. So you want to know the genome version. So in HumanMine, we only actually load one genome build and annotation version. Some InterMines load more than one in one InterMine. We don't put that specifically on the region search, but actually that's maybe something we could add. Now you've flagged that up. It will be in the data sources page, the genome build and annotation that we use. So hopefully, if I show you HumanMine, if you go to the data sources here, you can see the current genome build and annotation release there at the top. So hopefully that answers that question. Okay, I will go back to the questions. Right, the next one from Anne Nihus. Sorry if I pronounced that wrong. Is it possible, or will it be possible in the future to query metabolites or include metabolomics data? We get this question every single time, so I'm thinking we should probably include some metabolomics data at some point. It's certainly something we can look into. And if you have any ideas of specific data, then please do contact us and let us know what sort of data you would like to see, because we love to hear feedback from people and we like to know what people want in HumanMine. I mean, it's there for people to use, so please do let us know if there's any specific data sets that you would like to see in there. Okay, the next question is from Farid Musa. I could not find any microbial database in InterMine, particularly microbiome databases. As a suggestion, it would be extremely useful to access data in public resources like the DisBiome database, Earth Microbiome Project, or Human Microbiome Project through the InterMine interface or API. Yes, it would be. We did. There were some odd projects going on in microbiomes, but they haven't continued yet. Yes, that would be nice. Hopefully someone will get some funding one day to do that. So the next question is Claudia Pomarinki.
Yeah, she's clarified a bit better the question that she put at the beginning.
A little bit the expression level into account, in contrary to the, oh, I see, yes, I have seen that. We don't include that at the moment, but actually it would be really good to. And so at the moment we've just been sort of modernizing our interface a little. So if you come to InterMine in a few months' time, you may find the interface looks a little bit different. It's going to do everything that it does now, but it's just a little bit more of a modern technology behind it. And that's going to allow us to add more tools into the interface more easily. And that is something that I think we would like to include. So yeah, hopefully in the future we'll be able to add something like that. Okay, so I think that's everything. Okay. So I think now we're going to take a short break, give you all a bit of a breather before we move on to Daniela's API section. So the time now for me is 5:20. I know you're all in different time zones. So we'll take a break for 15 minutes, which means that we'll be returning at 25:02 and start again at 25:02, whatever hour you are in. Hopefully nobody's in half-hour different time zone.
We will put in the pop-up on the top. We will put the time in using the Central European time, which I think. Anyway, we return back in 15 minutes.
Yeah. Okay. Thank you.
maybe Rachel, you can stop the video and leave the share screen, I suppose.
Yeah, hang on a minute, I'm lost again. Gosh, it's so easy to get lost on your thing. Hang on a minute. I keep losing my screen. Okay, stop share. There we go. Ah, okay, I feel like I've babbled on for.
Hey, Rachel, you don't want to leave the slide with the coffee, so if the people, let me.
Yeah, should I bring that back up?
When you hear the ping is when I write it to the control room chat. Okay.
Oh, okay.
Okay. See you in 15 minutes, Rachel.
Yeah, okay.
Hi, Rachel. I think it's time to restart again. What do you think?
Yes, I think we should. Okay, I shall stop my share then, Daniela, and you can.
Okay. So welcome back, everyone. And so now I'm going to give you a short introduction about InterMine Web Services. I think it will last, I think, 20-30 minutes. And then we will move to the last part, which involve a practical exercise using InterMine Python package. So first, let me share the screen with you. Can you see it, Rachel?
Yes, that's fine, Daniela.
Okay. So we will start from a very short definition about what a web service is. Then I will move to describe it more about InterMine Web Services API. Then I will show you how you can access the web service, and I will end with, I think, a nice feature that we have in InterMine, which is the Python, or in general, any type of code generation, and I will show at the end. Before starting, I will start another poll. So the question is, do you know what a web service is? So I have started the poll, so please, a participant, click yes or not. I share the result. So at the moment, roughly, most of the people know what a web service is, but just a bit of us that doesn't know. So I will go a very quick definition. Thank you for participating to the poll. So I will end right now. Okay, so what a web service is. I have taken this definition from Wikipedia, partially from Wikipedia. I have a bit manipulated. So a web service is a server application which runs on a server, listening on a particular port, and listening for HTTP request. So when a web service receives an HTTP request, it can serve a web document. It can, for example, can be images, can be HTML pages, JSON file, or can create new resources, for example, or update existing resources, or delete existing resources. Any client which can make a standard HTTP request can make a call to a web service. So a client might require the creation of a new resource, or maybe the updating of this resource, or deleting an existing resource. An example of common HTTP client can be the browser, or if you like, a command line. For example, curl is another tool which allows you to make an HTTP request. When you make an HTTP request, you can specify the HTTP method. We have 4 HTTP methods: get, post, delete, get if the client wants to retrieve a resource, post if the client wants to create a new resource. Delete and put is for deleting a resource or for updating an existing resource. Where the web services are located inside the InterMine platform. Starting from the left, we have the Postgres database, where we integrate all our biological data. Then we have all the software, which is decomposed in several modules. So we have InterMine Core, InterMine BioCore, InterMine BioSources. All these modules implement different functionality inside the platform. And then we have this REST API, so the web services API, which is a layer. It's the interface between the functionalities provided by InterMine and all the clients that want to use these functionalities and want to display this functionality in a user-friendly way. Some example of clients which are using the web services at the moment, starting from the top right, we have BlueJeans. BlueJeans is the new interface for InterMine, which we hope to release before the end of the year. And the new interface is completely separated by the backend, and it only uses the web services API to communicate with the InterMine backend server. And other example of user interface are our two mobile apps. We have one app for Android, that one shown in the slide is the app for iOS system. And on the bottom right, we have the InterMine registry, which is an application, web application, which show all basic information about all the running instances of InterMine around the world. So I think having these web services is essentially a good practice from software technical point of view, because if at some point we decide to slightly change the implementation, maybe to improve or to fix a bug, we do not need to update different time. The web services are also useful when you want to access data programmatically without using the user interface. So maybe in some case you want to implement some script which allow you to automate database workflows, for example. Or maybe you can write programs which query different MIME instances. You write the program only once, you can run all the different MIME instances. These are just some, basic use case to show how useful the web services. So what you can do with InterMine web services? Actually, you can do all the things that Rachel has just shown with the user interface. I just put a list. This is not an exhaustive list. Just to give you an example, you can, with the web services, you can execute custom queries. You can run the template queries. You can search for keywords. If you are authenticated, you can create a list, and then you can rename. You can apply. You can operate. For example, intersection between list, difference, union. You can search for features, regions, and the last one, you can manage your user profile if you want. As I said, this is not an exhaustive list of what the web services provide. We have an extensive documentation here at Diadocs that I want to show you because I think it's very, very useful. So when you are here, you can select, I will select today Lyne. So here you can say there is a complete list of the web services. I can, for example, select the list. As I said before, you can see on the top right, get, delete, post, and put, the 4 different methods. So with get, you can have in return the list. Delete if you want delete a list, post if you want update, maybe rename a list, post, create, and put if you want to, maybe if you want to rename a list. So Diadocs is very useful because first you have a description of the web service. You can, if you want, you can execute the web services just to see if what you want. So the web services return the list, the public list created, publicly available in Lyne. Maybe you want to change a parameter in input. So for example, I will say change the format, and this will display HTML is returned, and I can see my list in a nice HTML format. So I return back to my slides. Okay, so when you call the web service, you need a URL. So the URL is composed by three parts. The first part identifies the MIME instance. For example, www.humanmind.org/humanmind/service identify the HumanMind instance. Then we have a second part, in this case it's search, which identify the functionality required. In this case it's the search. For the web service that I have just called was a list, for example. And my template, if you want to run a template, query result, if you want to run a query. And then we have the last part of the right, which eventually may be any additional parameter required by the web service. In this case, we are searching for the PPAG, so we need to pass to the web service. We need to pass the keyword, which is a PPAG. And if we want to maybe specify, this is not mandatory, the facet category, which is in this case G. Some web services might require authentication. So for example, if you want to create a list, you need to be authenticated. This is a basic example. If you want to have more detail about your account, you need to be authenticated. Or if you want to have in return the list that the user has access to, you need to be authenticated. So to call web services using authentication, you need an API access key. The API access key is very useful because it's much safer than using username and password. The API access key identifies in unique way your account, and it will last until you decide to regenerate a new one. When you create an account, by default, you don't have an access key token. You have to generate a new one. And I will show you soon how to generate an access token. But if you don't have an account, let's say you want to maybe play with HumanMind, maybe you want to create a couple of lists or stuff like that, you can create a 24-hour token, which will last for 24 hours. And in this way, you can save list without having an account. So I will show you how to generate an API access token. So I'm in HumanMind. I'm not logged in at the moment, so I log in to my account. Of course, if you don't have an account, you have to first create an account. And under my mine section, account details, here, I have my API access key. I can delete, for example, I can generate a new one anytime I want. Okay. So I return back. Okay, so I want to show you the option how I can access the web services API. So I can access the service API directly using the URL. So for example, I return back on my browser. As I said at the beginning, I can use my browser to make HTTP requests and so to call the web services. So an example, I call the web service list. I'm terrible in typing, so probably today So list, for example, I can see the list, the public list created in UMIM. I can change, for example, put format tab. I'm not sure if this web service, yes, I can open, and so I can open the result using a spreadsheet tool, for example. But if you like the command line, you can use curl. I'm not sure if you see my terminal. I zoom in, so I hope that you can see. Rachel, tell me if I have to zoom in better. So I can call my web services using the curl program, and I haven't returned the result. And another example of web service I want to show you was, for example, user OMI. Okay, so I run these web services and I have in return where my request must be authenticated. So to call these web services, I need to pass the token. So copy and paste it from here. And so now the web services return the information about me. Sometimes might be complicated call a web services using the URL, because in the example that I just showed was a simple example, so /list/user OMI. But if I want to run a query, it might be complicated, because when I run a query, the web services will be query result, I think. But after that, you need to pass the query. You need to pass how many items you want to return. You need to edit the format. So the URL might be very complicated. So we try to help on that, and we have this nice button, which is a web service URL button that I try to. For example, I want to run this template, and I have this web services URL here. So I can copy and paste the URL. As you can see, it's very complicated, it's very long. Maybe I can change it, maybe I don't limit it to 10. And the format is a tab, so I can open my result with a spreadsheet. Okay, so this nice feature, the web service URL, is not only on the template, but we have also in the query builder. So if you create a query, so let me create a very simple query. I don't know what to use, maybe publication. I can, from here, I have created my query, it's a very simple query. But I can, after I create my query, I'm happy with that, I can copy the web services URL. Okay, so let me see what we have next. Okay, so up now I show you a way to query our web services using a browser or using curl. But these are very simple examples. Often when you want to access, it's a good practice when you want to access a web service, to use the client library. InterMine provides client library for the most common programming languages. We have libraries for Java, Python, R, Perl. So you can query InterMine data using, for example, the library that we provide. And in some cases, in particular for template and for query, we provide the automatic code generation. So when you want to run a query, you first prepare the query in your web interface and user interface, and then after that you have built your query, or after you select your template, there is a nice button that I will show you soon, which allow you to generate the code in Python, in Java, in Perl, which language you have chosen. And this code will allow you to query the data programmatically. So it's a very simple three-step: choose, generate, run. And as I said before, you can apply on the template. So if you go to a template, you select a template, you can see a button on the bottom. You say generate the Python code. You say the Python code into a file, and then you run it. Or you can do the same thing with the queries. So let me just do an example. So for example, go back to InterMine. Maybe I want to start from a template. So we have two options, or you can do here, but I think it's not my favorite option. So this open a new tab with the Python code script. But I think my favorite option is to go and show the result. Then from here, on the top right, you say generate items by default. You can show comments or hide. And so this code has been generated. And if we run this code using Python, we will have the same result that the user interface shows us. So I will go into detail about what is a service, what is a query very soon in the second part of the presentation. Right now, I just want to show you that we save the script. It will save in a file called demo Python. Then I go back to my command line and I have already forgot how to call them, I think. Demo Python, yeah. Okay, so I have the same result that the user interface shows. So I think that I finished this part. Let me return back to my slide. Okay, so yeah, this was the last slide. So if you have any question, let me check on the OpenAir platform. I do not see any question about this part. If you have any question, please don't hesitate to add during the second part. So, okay. Okay, so we go back to the schedule, and yes, first another quick poll for you, and I will start soon. Which programming languages do you use? I have just started, so please put your option. Oh, a lot of people use Python. No, Python. Let me share the results, sorry. Okay, we have Python, 40%, R a bit more. Some of you Perl and Java. Nobody Ruby. Okay, so most of you are in R. Unfortunately, today I'm prepared to do the workshop in Python, but we can do the same stuff that we do in Python, we can do in R. Today I will, the practical exercise, we go through the same exercises that Rachel showed at the end. And this is a way to practice on InterMine Python package. I'm going to use Binder, which I will start right now. Binder is, I don't know if you know Binder, but it's a very nice tool. Actually is an executive environment which allows you to run Python without having any Python installed. Binder reads Jupyter notebook file. So we have prepared this exercise, this practical exercise in Jupyter notebook, and we run using my Binder. Usually, when we do this exercise, we are in a workshop where usually the participants are 20, 30 people. Today, it seems that we are much more participants. So I think that if you want to, please open your Binder and follow me doing the exercise. But we are so many, so I cannot stop and help you if you have an issue. In any case, in the agenda, there is also a link with the same exercise with the answer. So if you want to run later, please feel free to run later. So, okay, in this practical demo, what we are going to do? Just a few notes about my Binder. So my Binder, you can see there are some notes, some comments, some instructions. And then in the square we have the Python instruction. Then on the left of the square, you can see some square brackets. When you execute, when you are running instruction, you will see a star inside the square bracket. And then when the statement has been executed, you will see a number 1, 2, 3, 4. Okay. So this is just to explain how my Binder works if you try to do the exercise with me. What is the idea of the exercise? So we are going to do what Rachel has did with the user interface. So we are going to create first a list of genes belonging to the Paxt sick target list, which are expressed in the pancreas. So we are going to create a list. Then we are going to create a second list containing the genes which are associated with the disease diabetes. And then we will see, we will combine the two results and we figure out if these genes are associated with diabetes phenotypes according to genome-wide application study. So we start from the first query, and before creating a query, you need to instantiate the service class, and you need to import the InterMine web service module. When you instantiate a service class, you need two things. You need the URL of the mine that you want to query, and you need to pass the token because later in the exercise, we are going to create some lists. So to create some lists, as I said before, you need to be authenticated. So I go back to the human mine under my mine account details section to copy my API token And I will start to run, clicking run button. I will start to. So now I have a service class. When I have a service class, I want to query, I want to find the genes. So using the service class, I call the method new query. And because I want to query the genes, I put the root class, which is gene in this case. Now that I have the query, I need to set the view. So I want to set which fields I want to see, I want to have in return. To do that, I call the method add view, and I select the list that we have already put there for simplicity. Okay, so what I put in the view, I put a primary identifier symbol for genes, and then I put, we have, for example, level cell type attributes, which are attributes of the protein atlas expression class, which is a reference from the gene. I think that I can run this as well. So here my bundle tells me that the query has been located at this, I think is the memory location, I think. Then I want, so I create my query, I set what I want to see on my query. Now I want to add the constraints. So the first constraint, I want that my genes belonging to the Paxt 6 target list. To add the constraint, I need to call the add constraint method, where I need to pass the field which I want to constrain, which type of the constraints, and eventually the value. At the end, you see code A, which is in this case is not mandatory. So in theory, in a query, you will see soon, we can add as many constraints as we want. By default, when we add several constraints, we apply the operator AND. But if you want to change the logic, maybe you want to use a constraint one or constraint 2, you need to use the code A or B or C or whatever to apply a different logic. In this case, because we want to just operate to use the AND operator, we could not put the code. So let me execute this. So we have applied the constraints of gene. So we want all the gene belonging to the Paxt target list. Then we want to add another couple of constraints. So we want, I said before, we want these genes expressed in the pancreas. So to express these constraints, we need to add another couple of constraints. Typo. So the first constraint will involve protein atlas tissues name that we want to be equals to pancreas. And then I will add more constraint because we want the level be medium or high, of course. So we'll apply these constraints on the level attribute. The constraint that I'm going to apply will be one of. And then I put as array the value medium high. I was checking that I didn't put any typo before running, so I run, and then my query is ready to be executed. So I can call the method query.rows and print out the result to see if, okay, if what we wanted. So, so I go through every row and I print the primary identifier, symbol, cell type, level, reliability, and tissue name. Now, what I want to do is save the primary identifiers returned by this query and save into a new UMIM list. So what I'm going to do is I first create a Python list, which I call up in pancreas list. And then I want to append in this list, I want to append all the primary identifiers of the genes expressed in the pancreas. So I create a new cycle and I say up in pancreas, append primary identifier. Append, I forgot, I will run again. Okay, and just for a check, I print out the Python list to see if the Python list contains the primary identifier, and it does. So now we have this primary identifier just saved into a Python list. I want to save into a UMIM list. So to do that, we need to use a new class, which is the list manager, which provides a lot of functionality related to the list. So I create the list manager from the service. I call the method list manager. This will return the list manager. And when I have the list manager, I can create a list in UMIM. When I create a list, I need to pass three things. The first is the content of my list. In this case, the content will be the Python list just created. I need to pass the list type, which is gene. As I said, Rachel, before, we cannot create a list containing different items. At least not in InterMine. So, but we needed to specify the type, which is gene. And as a last argument, we put the name of the list that we are going to create in UMIM. I use the same name of in pancreas for simplicity. Okay, so let me check service list manager. With the list manager, create a list, content, list type, and name. Okay, should be okay. Okay, so a list has been created. Let me check in UMIM if we have the list. I go under my list. Here I can see up in pancreas, which has been just created, is a list of genes containing 129 genes. So we go back. So we have created the first query and saved the result into a list. Now we move to create the second query, which I said before. What we want from this query, we want all the genes which are associated with the disease of diabetes. So we run a new query, so service new query, and again the root class will be gene. We need to add a view. Probably this time we just want a few things. We want a primary identifier and symbol, will be fine. And we want to add the constraints because it's not enough. Otherwise, I will have in return all the genes contained, and it's not what I want to run now. So I want to add a constraint and say I want the organism name be equals to Homo sapiens. So organism name equals Homo sapiens, and then I will add another constraint. Maybe if I put it correctly, constraint. Okay. And then I will add a second constraint related to the disease name. Which the operator that we are going to use. We do not specify specific diabetes, but we just want to be generic. So we want the disease name contains diabetes. And then we execute this second query and print out a primary identifier and the symbols. I was checking if I was able to put any other mistake, but let's run it, maybe it's easier. Okay, no mistakes this time. So the query return primary identifier a symbol of genes associated with the disease diabetes. So we want to do a similar thing that we have just done. So we want to save the primary identifier in a new list. So I'm going to create a new diabetes genes Python list. And I will append the primary identifier into this list. Okay, and now I want to create a second list in HumanMine using the list manager. I call create a list method, where again I need to pass the content that I want to save in the list, which is diabetes genes. I want to pass the list type, which again is gene, and I pass the name of the list, which will be diabetes genes for simplicity. Okay. So again, create a list. Let me check on HumanMine if the list has been created. Yes, we have a diabetes genes with a description, list created with Python content library, type of gene, 60 genes. Okay, so last step is apply the intersection between these two lists. And the list manager provides a method for the intersection different union. So we are going to use the intersect method because we want to intersect the genes belonging to the two lists. So the intersect method receives an input, an array, where we put where I put the name of the list created in HumanMine, and I needed to pass the name of the list, probably, and I will call intersect the list. So let me run sorry. Okay, so now we have a list which contains only 3 genes. Let me open the list. Which contains these 3 genes. We can retrieve the list also using the Python library, we have a method which I think is called getList. Yes, it's getList, where you pass the name of the list, which is intersected list. And we save into a Python list. So let me execute this. Get list. Put. Then we run again. And let me print the result. So you say, yes, intersected the list, the gene contain 3 genes, the time and the description. So, okay, so probably I ran too quickly because, anyway, I do not see, before ending, I do not see any other question on Python or web services, so I will continue. And so the last part will be, I want to check, I want to see if there was any association of the 3 genes that we have just seen in the intersected list with the diabetes phenotypes according to the GWAS studies. So we are going to create the final query, and in this case, the root class is not gene anymore, but it will be the GWAS class. So I'm going to create a new query. I will add to the query some attribute to the view. In particular, I will add the result associated genes, and I will add the result p-value and the result phenotype. So I will add to the view all these fields. And now I have to apply the constraints to this query. So we need to apply 2 constraints, I think. So the first, we want first that all the associated genes belong to the intersected list just created. So this is the first thing that we have to put it. So the result associated gene need to be in the intersected list. Then we need now we want that the result phenotype contain diabetes for our query. So result phenotype, then the operator contains diabetes. And then we want to put a constraint on the level, on the result p-value, sorry. And for that, we are going to use another operator, will be minus equal this value, which is dot 0004. Correctly. Okay, so I think I put everything. So all the constraint associated gene belong to intersected list. I wanted the phenotype containing diabetes and the p-value minus or equals to dot 0004. So let me run this query. And now, as you probably already know, we run this query calling the method rows, and we print out the result. And maybe, because these are a lot of information, maybe we just print out the symbol Okay, from a quick look, we can see that we have only 2 genes which are associated to the disease of diabetes. So 2 genes out the 3 genes belonging to the intersection list. So I think that I end with the practical part. I want to show you another couple of links. I go back to the schedule. So here I put some useful link. For example, for Python, we have some nice Python tutorial. This is a repo, a Git repo, where we have some useful, simple Python exercises that will help you to familiarize with the Python package. And these are in the format of Jupyter Notebook, so you can run with Jupyter Notebook, or you can use MyBound if you prefer. Then we have this Python tutorial video, which has just been recorded during the summer, during internship outreach program, sorry. And these are very useful because, you know, in general, I don't like to follow a tutorial, but these are very nice because they are very short. And if you need a specific help about, you know, a specific thing, a query, template, enrichment, there is a nice title that we can help you to navigate, to browse on the different video. And I think that's very, very nice. And then, as a last part, we have the Python API docs, where you can find more information about how to use the Python API. And we have a similar thing in R, and we have some Jupyter Notebook, and which include the same workload that I just showed in Python. So I think I finished, and I over to you, Rachel. And first, let me check if there are more questions about Python.
There's a question about the API key, Daniela. Someone has registered with an ELIXIR account, but they don't seem to be able to create an API key. Created their...
Okay. So I cannot see. So I will have a look on that. So I'm happy first that you try to log in using the ELIXIR login. I will have a look on that. It might be a bug that we didn't realize. And sorry for that. And I will create a ticket just after the webinar. Yeah. Any other question? Because I cannot see, it's too small.
I don't think there's anything specifically related to the API. There are a couple more questions that are there. There's one from Maria about plans to include some disease data and some mouse phenotype data. So yes, nice suggestions. We do have plans to include the disease data. The mouse phenotypical data, I can pass on your suggestion to the MouseMine database, and or it might even be worth contacting them yourself to see if they would include the IMPC data that you're talking about. At the moment, we don't have plans to include that data in HumanMine. But, I mean, we perhaps could, so we can consider that. There was another one. The extent of isoform related information. So at the moment, we don't have an awful lot of isoform information. We load isoform proteins from UniProt, so you can always see what isoforms there are and get the sequence for protein isoforms. But we really don't have much data actually directly connected to specific isoforms. If you have suggestions, of course, we would love to hear them. Because that would be really, really nice data to add. So please let us know if you have any suggestions. There's another one. Oh, hang on, here's one for you, Daniela. is it possible to retrieve meta information, the data source and versions via the API?
I unmute myself. Yes, we have information about the core model that we have used to build. I just reshare my screen. Okay, so we have, for example, a web service which is a service model which return the model which we have used to create this specific instance, because in general InterMine is based on a common core model, but every instance can extend this core model, adding more specific classes. So when you run the service model web service, you have to return the model which has been used to create that particular my instance. And then we have, I'm not sure if I was able to answer your question, but we have another couple of metadata which are related to the version of InterMine. So we have a version InterMine which return the version of InterMine which has been used to create the build. Then we have the version release which is related to the data, and the version which is related to the version of API. So these are three methods, different web services related to the version. But you can find everything in IO docs. And any more questions that you can read?
I couldn't see any more.
Okay. Okay, but please feel free to send, or if you need more clarification, to send an email to our support mailing list. But I think Rachel will mention all our contact points.
Yes, I will just finish
so
up.
Let me stop my sharing so you can share this last slide.
Okay, so just to finish up, we've shown you an awful lot today, and so I'm sure you haven't remembered and taken it all in. So if you need more help, if you're anything more you want to know about, then we have lots of online help. So for the user interface, we have an extensive manual and videos linked under the help link on Flymine, but this manual does apply to all InterMine databases, of course. And then each individual InterMine also has its own help pages, videos and tutorials. Also on every InterMine page, you'll see at the bottom there's something called questions, comments. There's a link. So if you're using InterMine and you get stuck, then use that link. And that link is particularly useful because if you're actually trying to run a query or create a query, it sends us what you're trying to do as well. So it will send us the query that you've tried to run, so we can see what's going wrong. We have also a support chat, a support email. We're on Twitter. We love to get feedback. So any features or data that you want included in InterMine, then please do get in touch with us. You'll be sent out a survey anyway from the ECCB conference, but as we're part of the doing this under ELIXIR, we've also been asked to provide you with another survey. I've put the link here, which obviously isn't all that useful, but this will also be sent out to you. So please do provide us feedback for today's workshop. It's the first time we've run such a big workshop virtually, so any feedback will obviously help us improve things for the next one. And we've also been asked to advertise the ELIXIR virtual hackathon, which is happening in November. So this is a biohackathon. I think you have to Daniela, I'm sure you know more.
Yeah, we there with the 3 projects, and in particular, if you are interested in creating your own my instance, because you have some data, particular data you want to create, we might be there for helping you. So consider, and maybe it's better if you anticipate writing an email, so we can eventually prepare or to know a bit more about your data. But we have a project also open for this. So feel free to come and ask for help, and we will help you to create a new instance of InterMine with your data.
Great, thanks, Daniela. So registration for that is open until the 18th of September. Okay, so I think
it's free, of course, because it's free for us, so it's free.
So thank you, everybody, for listening to us today. We hope you've enjoyed the webinar and the demonstrations and have learned lots about InterMine. And obviously, we hope that you use InterMine after this. And as I've already said, please do feedback, or just let us know anything that you'd like added to InterMine. If you are interested in creating your own InterMine, or using the new InterMine, looking at the new InterMine in the cloud project, then also do get in touch with us. Okay, thank you very much.
Thank you, everyone. Bye.